Today our topic of discussion is going to be glycolysis and gluconeogenesis, which are the primary metabolic processes which are occurring in a human system or any living system. Before we come to the glycolysis process, let's discuss about how the glucose molecule enters inside the cell. We take glucose in the form of rice, wheat, whatever it may be, and then the metabolism or the digestion of glucose initially starts in the mouth or the oral cavity itself. The saliva has got several enzymes which has the ability to degrade glucose molecule, such as we have got amylase molecule which can digest starch. We have amylase to digest starch, which is present in the taste, which is present in the saliva. Similarly, the other digestive enzymes such as lactase, isomaltase, sucrase, which are the other enzymes which digest carbohydrate, are present in the intestinal lining cells or the villi of the intestine, where the absorption of the carbohydrate molecules also take place. Now, how the cells take in the carbohydrate, that is the most important part. There are certain receptors called the GLUT receptors, GLUT group of receptors which are present, which helps to take the carbohydrate molecules inside. There are GLUT1, which is present in almost all parts, all the cells of the body. We have got GLUT2, which is present in the intestinal cells. GLUT3, which is present primarily in brain. GLUT4 is one of the most important, and it is an insulin dependent glucose transporter. We have got GLUT5, which is a fructose transporter, which helps in the transport of fructose. We have got GLUT7, and we have got S-GLUT, which is a sodium glucose co-transporter. In this, the energy which is required for the glucose molecule is actually the energy is being acquired from the transport of sodium molecules. So first the sodium molecules enters, and the energy which is derived from this process is used in the transport of glucose. Now coming to the glycolysis pathway. Glycolysis is also known as the Embden-Meyerhof-Parnas pathway. So the glycolysis is a primary pathway which takes place in all living cells. It is also known as the Embden-Meyerhof-Parnas pathway. It is a process in which the glucose molecule is converted into pyruvate molecule. So it comprises about 10 steps. In the first, the entire glycolysis process can be divided into two parts: an activation pathway, and another is an energy generating pathway. From glucose till glyceraldehyde 3-phosphate is the activation pathway, and glyceraldehyde 3-phosphate to pyruvate is the generating pathway. Generating part of the, rather not pathway, generating part of the glycolysis process. It comprises about 10 steps. For the activation path, we require 2 ATP molecules. Whereas in the generating path, we get 4 ATP, but as 2 molecules are already being used up, we actually gain 2 ATP molecules at the end of the pathway. Now, coming directly to the pathway, the first step of glycolysis will include where glucose molecule is converted into glucose is acted upon by hexokinase or glucokinase. Hexokinase having higher affinity for glucose molecule than glucokinase. Hexokinase can act on glucose when the glucose concentration is as less as 0.1 millimolar, whereas glucokinase requires a higher serum concentration of glucose, which is about 4 to 5 millimolar. So when it's acted upon by glucokinase or hexokinase, it requires Mg2 plus ATP complex. ATP molecule gets converted to ADP, and we finally get product called glucose 1 phosphate. It's called the glucose 6 phosphate. Then on glucose 6 phosphate we add phosphoglucose isomerase. Then we get fructose 6 phosphate. When we get fructose 6 phosphate, now it is acted upon by phosphofructokinase 1, which will convert fructose 6 phosphate into fructose 1,6 bisphosphate. This is fructose 6 phosphate. This fructose 1,6 bisphosphate, once formed, the compound is directed only to the glycolytic pathway and cannot be transformed to any other pathway. So fructose 1,6 bisphosphate now is acted upon by an enzyme called aldolase, which converts fructose 1,6 bisphosphate into 2 compounds. This is known as dihydroxyacetone phosphate. This is known as the glyceraldehyde 3-phosphate. Now, this dihydroxyacetone phosphate cannot be used in the subsequent reactions. So, we have got an enzyme called triose phosphate isomerase. Triose phosphate isomerase, which converts this product, dihydroxyacetone phosphate, to glyceraldehyde 3-phosphate, so that this product does not get wasted. So, ultimately at the end of this aldolase reaction, we get 2 molecules of glyceraldehyde 3-phosphate. After we get 2 molecules of glyceraldehyde 3-phosphate, Now this is acted upon by glyceraldehyde 3-phosphate dehydrogenase. We get this is 1,3-biphosphoglycerate. It is 1,3-biphosphoglycerate. 1,3-biphosphoglycerate then gets acted upon by phosphoglycerate kinase to form glycerate. 3-phosphoglycerate then by a enzyme phosphoglycerate mutase get converted to 2-phosphoglycerate through an intermediate which is for 2,3-biphosphoglycerate. We get 2-phosphoglycerate. On 2-phosphoglycerate, subsequently we add phosphonol pyruvate, and in the formula which finally is acted upon by pyruvate kinase to form pyruvate. So this is the final product which we receive. Now, coming to the first step, glucose is getting converted to glucose 6-phosphate. We require an Mg2 plus molecule and an ATP complex for this process. And once this glucose 6-phosphate is formed, the cell, the glucose molecule which has entered the cell cannot leave the cell Glucose 6-phosphate is converted to fructose 6-phosphate because the hemiacetal position of fructose 6-phosphate is more prone to the second round of phosphorylation which takes place by the enzyme phosphofructokinase 1. So fructose 6-phosphate, this hemiacetal position is more prone to the phosphorylation by the subsequent ATP molecule. Here we have a transformation where ATP, another molecule of ATP, gets converted to ADP. We get fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is a product which, when formed, cannot get into any other pathways. Fructose 1,6-bisphosphate, when acted upon by aldolase, gives two products: glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Dihydroxyacetone phosphate is a base product and cannot be used by subsequent reactions. Therefore, it's acted upon by triose phosphate isomerase to get converted to glyceraldehyde 3-phosphate. Now we have two molecules of glyceraldehyde 3-phosphate. Then glyceraldehyde 3-phosphate acted upon by glyceraldehyde 3-phosphate dehydrogenase. In this step, we require an NAD plus molecule. This is a very important part. NAD plus molecule is required in this step, and we get NADP plus we get an NADP plus molecule from this step. Once the desaldehyde 3-phosphate form 1,3-biphosphodiglycerate. 1,3-biphosphodiglycerate is acted upon by the phosphoglycerate kinase. Actually, one more thing you have to remember, that is, in this case we have a phosphoanhydride group which is very important for the formation of ATP molecule in the subsequent step, because this phosphoanhydride group which is present is the molecule which means this compound will have a very high phosphorylate group transfer potential. It is acted upon by phosphoglycerate kinase to form 3-phosphoglycerate. This 3-phosphoglycerate molecule once formed, in this step we have got formation of an ATP molecule. Here an ADP molecule gets converted to ADP by the process known as the phosphorylate or substrate level phosphorylation, by a process called substrate level phosphorylation. Substrate level phosphorylation is nothing but transfer of phosphoryl group from in the site itself, from a compound having a higher phosphoryl group transfer potential to a compound having a lower phosphoryl group transfer potential. Now this 1,3-biphosphodiglycerate is getting transferred to 3-phosphoglycerate. So in this step actually 2 molecules of ATP molecules are formed, so thereby it is just counteracting the 2 ATP molecules which has been lost in the previous pathways. So this is just we just reached the break even in this part. Subsequently we proceed further. Now this 3-phosphoglycerate is acted upon by phosphoglycerate mutase to form 2-phosphoglycerate and then subsequently to the phosphenol pyruvate. This reaction is very important because this 2-phosphoglycerate molecule has a very low phosphoryl group transfer potential, so we need to get converted to phosphenol pyruvate which has a higher phosphoryl group transfer potential. So once this phosphenol pyruvate is formed, it gets converted to pyruvate. Here we again require Mg2 plus molecule, and also ATP is getting converted to ADP. ATP is again converted to ADP, thereby forming pyruvate. It's ADP getting converted to ATP, and therefore we have another 2 molecules of ATP getting formed, thus leading to the formation of pyruvate, and here also 2 molecules of ATP is formed, thereby the net gain in ATP is about 2 ATP molecules at the end of this pathway. But subsequently we see that we have got an NADP molecule, 2 molecules of NADP which is also formed in this reaction. After this NADP molecule enters the electron transport chain, we see 1 molecule of NADP gives rise to about 2.5 molecules of ATP. So 2.5 into 2 will give another 5 molecules of ATP. Therefore, total at the end of the glycolysis process, we have 5 plus 2, which is 7 molecules of ATP. Now, all the reactions of glycolysis are basically reversible reactions, except for this reaction which is 1, this reaction which is 3, and the last reaction which is 10. These 3 reactions are the only reactions which are irreversible. All other reactions are reversible reactions. So only these 3 are the irreversible ones. Now, coming to therefore, these 3 reactions are the reactions which are the most regulated steps of the glycolysis pathway. Hexokinase is regulated basically allosterically by the concentration of glucose 6-phosphate. Glucose 6-phosphate acts as an allosteric regulator of hexokinase. When the glucose 6-phosphate concentration is high, then the hexokinase molecule is inhibited, and hexokinase is also regulated by high ATP concentration. When the concentration of ATP is high, then also glucose 6-phosphate, this hexokinase molecule is inhibited. So it is inhibited by glucose 6-phosphate and by high ATP. Similarly, hexokinase molecule is stimulated by high AMP molecule, which is a depictor that the cell's metabolic energy is low. Coming to pyruvate kinase step, the pyruvate kinase step is again, this pyruvate kinase is again allosterically regulated by high concentration of pyruvate, high concentration of acetyl-CoA, which is a product which is formed subsequently from pyruvate by reaction with pyruvate dehydrogenase to form acetyl-CoA. So high concentration of acetyl-CoA is inhibitor of pyruvate. Similarly, high ATP concentration also inhibits the pyruvate kinase step. Pyruvate kinase is stimulated by high concentration of fructose 1,6 bisphosphate and also by high amount of AMP molecule, which is a stimulant which depicts that the cell's metabolic energy is not being met. Now we come to the most important step, which is the regulation of this phosphofructokinase step. The regulation of phosphofructokinase step is actually a reaction which is regulated by the two major glucose metabolizing hormone, glucagon and insulin. So, for this phosphofructokinase 1, we come to know that the allosteric regulator is a product called fructose 2,6 bisphosphate, which is formed with the help of enzyme phosphofructokinase 2 from fructose 6-phosphate. Therefore, we have to get a product called phosphofructokinase 2 is required. So, this phosphofructokinase 2 basically has two forms. One is a phosphorylated form or the inactivated form, and the dephosphorylated form, which is the activated form. So, for the conversion of phosphofructokinase to inactivated to phosphofructokinase 2 activated, we require an enzyme called PPP or phosphoprotein phosphatase, which is stimulated by an enzyme insulin. So insulin produces an enzyme called phosphoprotein phosphatase, which will convert or which will dephosphorylate the inactivated phosphofructokinase 2 to phosphofructokinase 1. And this phosphofructokinase 1, when activated, converts fructose to fructose 2,6 bisphosphate. And when this insulin concentration is high, when after a meal, when there is a lot of carbohydrate in our body. Similarly, when for prolonged fasting, when there is prolonged fasting and the glucagon level increases, glucagon stimulates a protein called protein kinase A, which phosphorylate the active form to inactive form. Thereby, it converts phosphofructokinase 2 from its activated form to inactivated form, and therefore inhibit the conversion of fructose to fructose 2,6 bisphosphate, and also stimulates the fructose 2,6 bisphosphatase activity, which converts fructose 2,6 bisphosphate molecules to fructose back to fructose. So this is a very important step. So that like glucagon stimulates the enzyme phosphoprotein kinase A, which converts phosphofructokinase 2 to phosphofructokinase 2 inactivated, and thereby this stimulates the fructose 2,6 bisphosphatase activity, which converts fructose 2,6 bisphosphate to back to fructose. Therefore, this fructose 2,6 bisphosphate is a molecule which is obviously required for the activation of the phosphofructokinase 1. Therefore, fructose 2,6 bisphosphate is an allosteric regulator of the phosphofructokinase 1. So, this fructose phosphofructokinase 1 is stimulated by fructose 2,6 bisphosphate and by low ATP content or high AMP. Similarly, it is inhibited by high concentration of citrate and high concentration of ATP molecule. So this is the overall derivation part of glycolysis.